Okay, so just to summarize what we went over, this is looking at the coronavirus, the SARS-CoV-2. These are the S spikes, and like we said, what'll happen is this will go into, you breathe it in, it goes into the lungs, and then from the lungs, what it will do is it will attach to something called the pneumocyte. So there's two types of pneumocytes. This is a pneumocyte in which we do gas exchange between the lungs and the bloodstream. There's gas exchange here, okay? So what's going to happen is you have the ACE2 receptor, and my virus is going to attach onto the ACE2 receptor. When it attaches onto the ACE2 receptor, it's going to be brought into the pneumocyte, and then what will happen is you can see it's covered here in the membrane. This is called endocytosis. It's covered in the membrane, and if you notice, we have the RNA inside there. What this will do is it will release the RNA, and then the RNA will go to the ribosome. So this here is the ribosome, and it will use that to make proteins. Or proteases, polypeptides. It's going to use it to make polypeptides. So here's my polypeptides. Then proteases will now turn those polypeptides into things that these virus can use. Okay. The other thing that's going to happen is we're going to make some RNA-dependent RNA polymerase, and what this will do is it will cause the RNA to make more RNA. And so once that happens, now we have the RNA, we have the spikes and other things that are needed for the virus. Those proteases will put those together, and we are going to end up with a new virus. The Golgi apparatus will then package that, and it will leave the cell.